The overall goal of this experiment is to characterize DNA aptamer small molecule interactions, including binding site mapping, using microscale thermophoresis.
This method can help to answer key questions on basic binding parameters of molecular interactions. The main advantage of this technology is that it is independent of interaction partner size, allowing quality control data of challenging interactions between aptamers and small molecules to be obtained.
Though this method can provide insight into aptamer-small molecule interactions, it can also be applied to other molecular interactions such as drug-target or antigen-antibody interactions.
To begin the procedure, prepare a 100 micromolar stock solution of the Cy5 labeled DNA DH25.42 aptamer in distilled water. Then, dilute the aptamer stock solution to 200 nanomolar with binding buffer. Incubate the aptamer working solution for 2 minutes at 90 degrees Celsius. Cool the solution to room temperature on ice. Next, label and set out low-binding 200 microliter microcentrifuge tubes for a 16-step serial dilution of the ligand to be tested. Pipette 20 microliters of a 10 millimolar stock solution of ligand in binding buffer into tube 1. Pipette 10 microliters of binding buffer into tubes 2 through 16. Then, transfer 10 microliters of ligand stock from tube 1 to tube 2